Okay, so I will let you be my guide when myself and my colleague, Dr. Greg, can start.
Actually, I think you can just start, sir. I mean, we're
Okay.
216
Okay.
already. It's good to
Okay.
be
Good. Good. Good.
precise like the
So
German.
let me just give a very brief introduction, 2 minutes, and then my colleague, Dr. Laura Gregg, will do a little presentation. After which, I will then have a set of slides of myself. So myself, as previously discussed in a previous call. So I'm Joseph Borch from Malta. I've been working now roughly in this field for just about 10 years, maybe a bit more. So even my previous years doing the master's degree and the PhD, and eventually spending some time also with Jacques Philipsen in Holland. So the work was always focused on blood and very specifically on thalassemia research and sickle cell disease, because the defect, the problem is on hemoglobin basically. So mutated or abnormal genes that would lead to abnormal or a deficient level of hemoglobin. So we work on this topic using various tools and ways and techniques, as you will see in the presentation. We pride ourselves on using both basic and fundamental type of genetics. So a lot of tools that are available to us in the lab, but we are also lucky enough to be doing a lot of clinical research. So we also have access to patients, clinical patients that attend clinic weekly or monthly. And because of this, we have a database, a repository of patients that we can follow up and also invite to participate in our projects. Of course, ourselves in Malta, there's a very small island population. We have roughly around 40 patients that are thalassemia, not so much sickle cell disease, although we do see some from migrants coming or hailing from North Africa. I am aware that our colleagues in Holland, there is a larger cohort of sickle cell anemia patients. And we also have our colleagues in Germany, in Heidelberg, who, despite not having too much of clinical material, we have access to their facilities, which sort of screen hundreds and thousands of compounds, and also we can make use of next generation type of tools and technologies. So as an introduction, Laura Gregg, so she finished her PhD roughly a year ago, if I'm not mistaken, or a bit more. And she now works full-time, specifically as a postdoctoral researcher with myself and my group. So she is based at the University of Malta, and she works specifically on the same topic of blood, hemoglobin switching, thalassemia, and sickle cell disease research. So what she will present today is a large portion, not all, a large portion of the work that she conducted on the Maltese patients and from the Maltese side story point of view. So I will let Laura commence her presentation. I believe she will share screen for the slides.
Okay. Good afternoon. How is disabled participants screen sharing?
Okay, so we need to give access to screen sharing to Laura Gregg. If you can, Emmanuel or Oludare.
Laura, you should be able to share screen.
Okay, Laura. Okay.
Okay, thank you.
Yes, we can see your screen.
Okay, so today I'm going to talk about my PhD work, which was mainly focused on KLF1, which is a gene, as we will see later, on chromosome 19, and I will talk about the role of KLF1 variants on hematological parameters and their significance to hemoglobin disorders. So the KLF1 gene is located on chromosome 19, and it consists of 3 exons and 2 introns. And it has three zinc fingers, which are represented by F1, F2, and F3 in this figure, which enables the KLF1 to bind to specific sites in the genome, typically to the CACC boxes in the beta globin and also to GC-rich elements. KLF1 activates a diverse set of genes that have an important role in the regulation of key pathways, as we can see, and therefore mutations in the KLF1 gene can give rise to a wide range of hematological disorders. In 2010, Prof. Borch identified a large family with hereditary persistence of fetal hemoglobin. So mainly in Malta, we have a screening program, and we screen every newborn and every expecting mother, and we screen them for thalassemia. And what happened, this family presented with an HbA2 and an elevated HbA2 and an HbF between 3.3 and 19.5%. And after carrying out DNA sequencing, two mutations were identified in the KLF1 gene, and there was a nutri substitution, and there was the K288X mutation, which was a truncation mutation. And after carrying out functional work on HEPs, human erythroid progenitor cells, from these subjects with high fetal hemoglobin and controls, it was found that the KLF1 has a dual role. It activates the beta globin gene at the expense of the gamma gene by interacting directly with the regulatory elements in the beta promoter. In doing so, it represses gamma, and it also activates the expression of BCL11A, which in turn also represses gamma globin gene. So in figure A, we're seeing a subject with a normal KLF1. So you have elevated beta globin and downregulation of gamma, but in subjects with the K288X mutation, this balance is perturbed, and therefore you have an increase in gamma globin. There were a lot of studies done to see the effects of the KLF1 variants, and in fact, KLF1 variants are now classified in 4 classes. You have class 1, where you have variants with no or minor functional consequences. You have class 2, which are usually innocence variants or small unframed deletions, which interfere with the normal function of KLF1 gene. You have class 3, which are usually truncating loss of function variants, such as the K288X mutation, and you have class 4, which are dominant variants. And also, the effect of KLF1 variants on hematological phenotypes ranges from the in blood group. You can have variability in the HbA2 levels, variability in fetal hemoglobin levels, and also can give rise to elevated zinc protoporphyrins and also congenital dyserythropoietic anemia. So some of the objectives of this study were to explore the occurrence of KLF1 mutations in undiagnosed subjects with borderline A2 and beta thalassemia heterozygotes, and also to investigate the clinical and quantitative variability of fetal hemoglobin in subjects with the same KLF1 truncation mutation, but different fetal levels, as we will see later on. So, as I already said, and Prof. Borch said, we have, through the thalassemia clinic, we have this sample collection, and we collected 426 samples with an A2 between 3.1 and 6.7%. We performed beta globin sequencing, and according to their beta globin sequencing results, we divided these into either pseudothalassemia or else beta thalassemia heterozygote. So the pseudothalassemia are subjects that, although they have a clinical picture of a beta thalassemia heterozygote, so they have low MCV, low MCH, and an elevated A2, they did not have any mutations in the beta globin gene. And we also included 200 subjects with a normal CBC as our control group. And we did sequencing of the KLF1 gene. And also we found other family members with this truncation mutation, but they had lower fetal hemoglobin levels than the first family described by Prof. Borch. And we performed culturing of human erythroid progenitor cells at SANG in collaboration with Sangmin and Erasmus MC. And we also performed whole genome sequencing, and we performed also some promoter functional assays. So this is some hematological data. So as you can see, the controls, the average of their fetal hemoglobin, MCV, MCH, MCHC falls all within the reference range. But that of the pseudothalassemia, as you can see, that the MCV, MCH, and MCHC is lower than that of the reference range. And their average of A2 was about 3.3%. So in brief, we found 13 different KLF1 mutations. Most were either on the promoter region, on exon 2, or else at the 3' UTR. We found no mutations in exon 2, and these mutations were present in 55.3% of the pseudothalassemia group, 40.5% of the beta thalassemia heterozygote group, and 38% of the controls. So then we came across other 5 families who also presented with low MCV, low MCH, normal or slightly elevated fetal hemoglobin, and elevated A2. But when we performed beta globin gene sequencing, we found no mutation in the beta globin, but we found the same K288X mutation in the KLF1. So here we have the family trees of these 5, with these 5 new families. And as you can see, usually the proband is an antenatal mother who presents at the thalassemia clinic after performing screening for thalassemia, heterozygote for thalassemia. But these families, these mothers were found to be K288X. They had the K288X mutation. And this is the last family which we found. And as you can see, they all presented with low MCV. She had an A2 of 3.4, an elevated fetal hemoglobin, and we also did the F erythrocytes using flow cytometer. We carried whole genome sequencing on some of subjects of these families to see what's the difference between the first family with high fetal hemoglobin and these families that, although they had the same truncation mutation, they had different levels of fetal hemoglobin. And what we found that subjects with high fetal hemoglobin had 2 mutations in the promoter region of the KLF1, while the other subjects with medium fetal hemoglobin had 1 mutation in the promoter of the KLF1. So we had 1 promoter mutation, which was in cis with the K288X mutation, and the ones with high fetal hemoglobin, they had a mutation in the promoter on the other allele. So what we concluded from this research, we concluded that KLF1 mutations can be classified as autosomal recessive. In pseudothalassemia, KLF1 compound heterozygotes or homozygote mutations gave rise to borderline A2, low MCV, and low MCH, and also can give rise to an increase in fetal hemoglobin, while KLF1 mutations in beta thalassemia heterozygotes resulted only in more elevated HbA2 without any other clinical or hematological effects. And here we're seeing the importance of testing for KLF1 in these subjects that although they have a picture of beta thalassemia heterozygotes and they have no mutation in beta globin, you perform KLF1 sequencing because at least you can give them a diagnosis. The variability in fetal hemoglobin in subjects with the same truncation mutation could be counted by the allelic variation of the wild-type KLF1 allele. So what our hypothesis is that in a normal subject, in a subject with no mutations in KLF1, you have both alleles which are functioning. But in subjects with medium increase in fetal hemoglobin, you have one allele which is defective because you have the truncation mutation, but the other allele is functioning well. On the other hand, subjects with high fetal hemoglobin, you have one allele which is not functioning because you have the truncation mutation, but the other allele is also affected because you have the promoter mutation. And also we're seeing that our hypothesis is that KLF1 needs multiple hits to be affected. And this, as I already said, highlights the importance of KLF1 sequencing in patients with hematological features resembling beta thalassemia or other hemoglobin-related disorders for differential diagnosis of microcytosis. So this is more or less, in brief, some of the work I did for my PhD. And also we're working on other techniques in collaboration with Erasmus MC and Sanguine to do RNA sequencing and data sequencing. Thank you.
Thank you, Laura, for that. Okay, so this was a bit focused on a particular gene of interest that it has been well studied in the past, but proudly we say in the past 10 years, so in 2010, ourselves and together with our colleagues in Holland, we had identified that the gene KLF1, when perturbed or mutated, it actually influences or affects hemoglobin production, specifically fetal hemoglobin upregulation. Now, high levels of fetal hemoglobin is something which is desirable in people with sickle cell disease and thalassemia. Because sickle cell disease and thalassemia, the problem, the issue is the adult hemoglobin. It's abnormal, it's defective, and it provides a problem. So if we manage to tilt the balance and increase fetal hemoglobin, which is presently innate in very, very low levels, but if we can switch it back on to high levels, that can be a curative or an appropriate treatment. So now I will build on what Laura Greg was talking, and I will move on to our next set of slides, which is a joint project, beautiful project between ourselves in Malta, in Holland, and in Germany. So let me see if I can share screen as well.
Okay, so if I can summarize a little bit what you've been saying so far, what Laura just said. I can see one of the executives of the Nigerian Association of Nigerian Nurses, Mrs. Oti Akimbo, is incidentally joining us. What you're saying is that fetal hemoglobin in high concentration reduces the chances of the expression of sickle cell and thalassemia.
Precisely.
I mean,
precisely.
I'm trying to speak in a lay term. That's
it. That is exactly it.
Exactly. But the fetal hemoglobin levels continues to reduce. That's why it's called fetal anyway. It's abundant in the fetus, but it reduces as the fetus becomes older, as the baby gets older. And that's why you often will have that expression. So if you can find a way to switch on the production of that hemoglobin on a gene level, then you'll be able to actually reduce the incidence of probably cure thalassemia and sickle cell. So you don't have a molecule or something that I can't, my biochemistry bits are still, something like a promoter of, sorry, a molecule that will enable the switching on of that particular gene since it's already identified. Okay, thank you.
Exactly. Of course, there are different ways how to approach the augmentation or elevation of fetal hemoglobin. So in the previous presentation by Laura Gregg, it shows you that KLF1 is one very important molecule that, or gene, that if you manage to suppress it or keep it low or switch it off, it will favor high levels of fetal hemoglobin production. And we've seen this in the lab, and we have seen this clinically. So those were the families that Laura was showing, the family 1, and then 4 more families. Those are people who have a mutation in KLF1, and when that is mutated, it affects fetal hemoglobin production. Now, to date, from a clinical model only, what leaves to be desired is, even though they have a mutation in KLF1, not everyone exhibits high level of fetal hemoglobin. So the mechanisms are a bit more complex. And we want to understand the full mechanistic detail to be able to pinpoint and design the appropriate molecules or treatment.
Yeah.
Now, what I will be talking about is on the same concept, but using a different approach. So let me see. Do you see this?
It's, yes, it's visible.
Okay. So I will talk about, a particular novel approach that we are preparing as a part of the treatment of sickle cell disease, not only because thalassemia will also be affected with this. But speaking of which, sickle cell disease is very, very interesting for us, and also from a geographical point of view, because we are aware, and I think the numbers might be even higher, but we are aware that roughly between 4 or 5 million patients have sickle cell anemia as an active disease globally, and much more than that, roughly about 50 million patients are carriers, have a sickle cell trait, which means that these are people who are carrying the disease, and there's the possibility of passing it on to their next generation. 80% of the world's sickle cell population is localized in the African continent. So whatever we discover and whatever treatments one will develop for sickle cell anemia, a large portion or the share of the market or of the treatment options will, of course, inevitably focus on the African region, because 80% of the patients are found in that region. So just a very 1 or 2 slides intro about Malta. So Malta is very, very small. So we have one main university, and we do a fair amount of R&D, research and discovery. Of course, being one university, it is not a specific university. So we also teach or educate people to become teachers, pharmacists, other healthcare professionals, architects. So we have all the fields. Of course, there are the fields that are life science research, medical research, biochemical, and that is the area that myself, I work in together with my colleagues. We are very lucky because for the small size of Malta, the university is located 5 minutes by walk from our national hospital. We also have only one national hospital in Malta, which is public, which is government owned. And then there are a smaller number of private hospitals. But having university and hospital next to each other gives us many, many advantages. And some of which, as you can see there in yellow, is of course the topic of haemoglobin variants, beta thalassemia, sickle cell disease. So much so that we have developed, and it has been running for the past 20 years, I believe, maybe a bit more. We have a program, it's called the Newborn Screening Program. And every single newborn in Malta, the blood sample passes through the lab where I work in, and it is screened for hemoglobin variants or disorders. So a large portion, 97%, 96%, are of course all normal, but a small percentage, a small fraction, we would identify hemoglobin variants, some of which might not necessarily pose a problem or a risk. They're just variants. There are thousands of such variants globally. But we also identify thalassemia traits or carriers. And of course, occasionally we also find sickle cell traits and some other very rare forms, especially when people come from abroad, from neighboring countries. So having the patients coming to us from hospital, having a national screening program gives us a lot of advantages. And we have learned a lot over the years how to make good use of screening programs. Because once you do that, you have a collection of samples that you can put away, safe away in a bank, in a biobank, and then you can also conduct research or recall back patients to do further research as we go along in the active program. Okay, so intro, you've heard Laura as well, maybe just a few pointers of thalassemia, sickle cell. That right-hand side photo is a typical thalassemia child who is not transfused. So he is, how do you say, he's not having active treatment. So thalassemia and sickle cell are some examples of classical genetics. I mean, classical means that the parents both have a mutation. So they are both carriers. We call it autosomal recessive. And therefore, in a recessive manner, their offspring, their children, there's a 50% chance that the child will be exactly like his father or his mother. There's a 25% chance that the newborn will be completely unaffected. So he inherits the portion that is normal in the father than the mother. And of course, there's a 25% chance that the newborn will inherit both mutations from his parents, the father and the mother. And that will provide a problem, and the major effects will be more pronounced and visible. On the right-hand side, you can see some typical normal or healthy red blood cells. They're these funny shaped, donut-shaped cells that carry large oxygen molecules by means of hemoglobin. And on the left-hand side, you can see how abnormally shaped, smaller and different shapes and sizes of shapes, including, so this shows of a thalassemia patient. Of sickle cell, they have a very peculiar shape, which are sickle in shape. You will see later on. Sickle cell disease, so we can focus a bit more on this, since this is of a much greater importance and concern in Africa. So if you had to get a lens and look at the hemoglobin genes, and you look at beta globin, so this is the portion of the gene that when it is normal, there's no problem, and hemoglobin is normal, both fetal and both adult. And there you can see those funny letters. Those stand for amino acids. Okay, they're parts of the building blocks of protein molecules. And you can see there in orange the glutamic acid, and the DNA sequence is GAG. So that's the DNA, and glutamic acid is the amino acid. The bottom layer, which is exactly the same, except for one other amino acid, which is valine. And this is because there is one DNA nucleotide change. So from all the DNA letters, there's one change, and that one change shifts or changes an amino acid from glutamic acid to valine. And that change is enough to produce visibly the condition of the disease. Okay, so normal hemoglobin A is a tetramer of 2 alpha and 2 beta, but sickle cell hemoglobin, the beta globin is mutated, and it is therefore abnormally shaped or structured. Of course, sickle cell disease will arise in patients who have both genes being mutated, meaning, like I showed you earlier, if this child inherits both from his parents, the adult hemoglobin of this child will be completely sickle globin. Okay, so thalassemia and sickle cell disease are also known as monogenic diseases. Okay, monogenic meaning we know exactly what the problem is. We know specifically that the defect or the gene problem is the beta globin, so we know that. But there are many pleiotropic effects. So how one patient presents himself or herself in the clinic might not be the same as another patient who presents himself or herself to the clinic. So there are these differences. So the primary effects are sickle hemoglobin polymerization. So you get sickle cells attaching to each other. You get sickling of red blood cells. So you can see an example here in this shape, blood film. Of course, if the cells are abnormally shaped, the body will try to eliminate these cells. So there is a hemolysis or lysing of the red cells. And of course, that would result in anemia. So the hemoglobin will start to go down. But that is not all. There are secondary effects as well. So as you might know, there are vascular damage, there's inflammation and cell activation. There might be also multicellular adhesions present. And finally, and very importantly, there might be vaso-occlusive events that can cause organ or multi-organ damage, such as the well-known stroke in brain or other organs. So living with sickle cell disease is by no means very easy or very simple. Okay, so there might be recurrent blood vessel occlusions. This might result in deprivation of oxygen and progressive organ damage. There is a chronic hemolysis. Okay, if the patient is not well followed up, the patient can be anemic. He can have jaundice, he can have gallstones and blood vessel damage. Again, if left unchecked, there might be neurological complications, including stroke. There's a very high risk of thrombosis in these patients. And therefore, based on these points, we are aware that there's a clear need to develop safe and effective treatment modalities for sickle cell disease. This has also been said by the FDA. There are a number of drugs and molecules on the market. In fact, hydroxyurea has been approved by the FDA, even early since 1998. We are aware that it's not the holy grail. It's not completely effective, okay? It reduces the frequency of painful crisis, and it reduces the need for blood transfusions, but not every patient responds greatly to this drug. This is also used in certain types of hematology cancers. Okay, so the rationale of ourselves, of our group, as you have seen in the previous presentation, HPFH, okay, the hereditary persistence of fetal hemoglobin is a real thing. It exists. So people with high levels of HPFH is protective. And if these high levels are present in sickle cell and thalassemia patients, this can putatively cure them. So we are after an induction of fetal gamma globin, and we achieve this pharmacologically by means of drugs and chemicals and small molecules. So right now at hand, together with our collaborators in Germany, we have a number of small molecules and chemotypes that we identified and are able to induce gamma globin. So this gives a very good starting point for the development of novel therapeutics. There are many characteristics that we want to abide to. Of course, we are under the idea that the drug, the molecule, should be orally available. The potency, okay, the concentration of the drug at which it elicits a response must be of a low dose, okay, not to have any major complications. We are after a gamma globin induction of 20% minimum or more, okay? And of course, we want to, okay, once we have these compounds at hand and we know that they induce high levels of gamma globin, of fetal hemoglobin, now we can determine functionally what's happening inside the cell, okay? So what is happening at the RNA level or protein level, okay? Why is the cell responding to the drug and augmenting fetal hemoglobin, okay? So through functional work, we can decipher or identify what's going on, and it's like clockwork or a puzzle, okay? Which puzzles are needed, which parts are needed to elicit this response. Okay, this is the premise or the rationale that we are working with. So as was suggested earlier before I started the presentation, so fetal hemoglobin, yes, it's present in very high levels in a fetal environment, so before birth. Okay? Now the phenomenon, okay, is that just before birth, okay, a few weeks before birth, the fetal hemoglobin starts to decline, and this is replaced by adult hemoglobin. Okay, the red line, it's unlabeled, the red line. And that's where the onset of symptoms for sickle cell patients begin. Okay, so our idea is that this blue line, the fetal hemoglobin, we want to two things: either stop it from declining or decreasing, at least to that gray threshold area, or else, in the case of adults, okay, we want to induce back high levels of fetal hemoglobin. It might be a bit more challenging, but that is also in mind. Okay, so the methods that we use is that we encounter the patients, we talk to them, we consult them, we invite them to participate in our projects, be it healthy adults and be it patients who have thalassemia and in the future also sickle cell patients. And from the blood that we obtain, we manipulate or process the blood to obtain specifically the early blood cells. We call them early erythroid progenitor cells. So these are the cells that, given the right conditions, they will grow, they will divide, and they will produce hemoglobin. Okay? So we want the cells earlier on so we can manipulate or add the chemicals very early on before they are committed to fetal hemoglobin or to adult hemoglobin. And what you're seeing there is a set of plates or cell culture wells that we use to plate these cells. Okay, so on the left-hand side is a typical photo of the cells that we obtain. On the right-hand side, you can see a white cell button. Okay, so those are the cells that we want to obtain for further work and experimentation. A white cell pellet means that they are still not producing hemoglobin. So that's good. We can manipulate the cells at that stage. It is no point or no use to manipulate cells if they are already producing hemoglobin. You want to go one step earlier, okay? Yes, so once we do that and we have the cells at hand, we grow them under different conditions. In brief, we have cells that are growing and growing, proliferating, and we have the same set of cells that we differentiate them to produce hemoglobin, okay? So we have two sets. Once the experiment is done, 48 hours, sometimes even 96 hours, okay? Once the experiment is done, we stop the cell cultures, we obtain the RNA from the cells, and we conduct our experiments. One main tool that we use is real-time PCR or qPCR. So basically, we want to measure hemoglobin, and of course, we measure the fractions. We measure beta globin, gamma globin, alpha globin, and all the others, okay? If you remember well, beta globin is the one that is defective in sickle cell, and gamma globin is the one that we want because it induces high levels of HbF, okay? So this is a typical example. I will not go too much detail, but on the lower left-hand side is a typical result of how the PCR progresses. Okay, so if there is globin RNA, it will be amplified and it will show. Okay, and on the right-hand side, this one peak shows you that you are amplifying correctly. Because if you get many different peaks, the experiments are very bad, and then they should not be used for analysis. Okay, that's very important. This is only a small set from many other results that we have. So this is data obtained from Malta, from my lab, but we have similar or even better data in our colleagues in Holland. And you can very clearly see that certain drugs or the molecules that we have at hand right now, with the different experiment profile, doing different concentration gradients, we can see a very, very favorable response in gamma globin and not so much in beta globin. So the chemicals that we have at hand seem to be working very much in the gamma program, okay? So all in all of this is a very good starting point, we would call it, okay? We are no way near in producing this tomorrow or next month, but this is a good starting point to continue our R&D, which holds a lot of promise for development of novel therapeutics. So I'm almost done. Yeah, so of course, as you might imagine, this is not a one-man project. It entails sharing of resources, and maybe hopefully in the future, the fact that we are talking or collaborating with African groups, we might also expand the collection of material. So we are after a compound that is very efficacious. We want to explore this in a disease model. So we have some specific animal models that we want to look at, or even cell models that we want to look at. Of course, we are aware that it must be an acceptable side effect profile. Okay, it can induce very high levels of fetal hemoglobin, but if it has a very bad side effects, then that is not good. And we are after something which is sort of orally available. That would be very, very easy, or something very useful on a mass scale. So right now, we are extremely interested and working hard to identify molecular targets, the mechanistic understanding is doing, and also our colleagues in Holland and Germany. And we want to determine the response of patient samples for the time being ex vivo. So we have patients, we take their blood, and we experiment in the lab. When we are satisfied that in the lab we have desirable results, then we can plan for the next steps of proof of concept or early preclinical studies. That is our ambition and intention. Yeah, this I think you might have, I said this in the very beginning. So within the sub-Saharan African, there's a large number of patients, 5 million or more. And then in different areas of the world, there are less. I think then the next high population of sickle cell patients is in India. And then the further west you go, there are less and less. But it still poses a problem, and one needs to tackle the situation in every respective countries. So last slide. It seems that we are aware that sickle cell anemia has been with us for a large number of years, and it will continue to grow. So there is a need which right now has not been put on hold, or there's a significant unmet need. Okay, we have at hand good starting points. So we don't have end products, but we have very good starting points that we want to develop further. Okay, we have a clear plan, especially by my group and others in the field, another of which is in our group today, Jacques Philipson. So all in all, myself and others, collectively, we have experience in this field. And finally, with the proper resources, be it budgetary, monetary, and also access to patient material, we can plan more precise, definitive experiments, such as animal models, proof of concepts, and this will definitely facilitate us to those participants in trials in the future. So that is the final concluding remark. Thank you. I hope I was clear.
Very, very clear, Professor Borg.
Okay. So
do we have anyone who wants to maybe give a comment? Jack Philipson, you have anything?
Yeah, maybe Jacques can add something from his experience and from his lab in Holland.
If I can, yeah.
Yes, sir.
Sorry, I'm struggling a bit with the technology. There we are. I can put the video on.
Okay.
Yes, so I think the aim of this research project is to develop hopefully a cheap a drug to treat the sickle cell patients, and that drug, the basis of that drug would then be that it reactivates the expression of the fetal hemoglobin. Now, you may be aware of other initiatives where people use gene therapy with a similar purpose. But the problem with gene therapy is that it is really very expensive. And I calculated even for the Netherlands, where we have 1,500 sickle cell patients, if we have to pay €1.5 million for every single patient, that is not feasible. So that is never going to work. And of course, worldwide, this will never be a solution. I also think that having a drug would be favorable, because you can adjust the doses of the drug. You can, if there's any inadvertent side effects, you can stop taking the drug. Well, then of course you're back with sickle cell disease again. But there's more flexibility. And at the moment there are no good HbF-inducing drugs available, but we know a lot more about the repression mechanism, as Laura told you about KLF1 and the BCL11A, and there's quite a large number of factors known in this network now that repress the fetal hemoglobin. So we really hope that we can not only develop a compound that reactivates the expression, but that we also understand how it works. And if we understand how it works and which proteins it targets, then that will allow to design better and more specific variants of the drug. So that's perhaps very broad what I'm telling you, but that is what we are aiming for. And I think it's very exciting to do this together with Malta and AMBL, because we have our own expertise. So I work in an academic hospital. We have in the Rotterdam area, we have about 600 sickle cell patients and a clinical expertise center. So that means we can test the drugs not on the patients immediately, but we can grow cells from the patients and test the drugs on those cells. Joseph, on the other hand, has these patients with beta thalassemia, which of course is also due to the beta globin gene, but then rather than a pathological variant. It is lack of expression of beta globin. So we can use that, and they have their Malta biobank, so they have cells frozen that can be used for testing. And then the AMBL in Heidelberg, they have this compound library that we will use in the screen to well, in first instance, to discover not the drugs, but more like the predecessors to these drugs. So get some hits in the library, then take these hits and optimize these for use as drugs, because the medicinal chemists can make all kinds of variants of these initial hits, and then you have to look at toxicity. Is it taken up by the cell? An oral compound would be preferable, of course, and so on. So that is drug development. So I'm rather excited about this project, and I really hope that we will be able to identify positive drugs, and we already have found one by well, that's really a coincidence. That is already a lead for further optimization by medicinal chemistry. But that is even before we start screening the entire library. So that is, I think that is what I would like to add to Jozef's.
Yes, I have nothing to. So that really summed up nicely what we are doing right now. So yes.
Thank you very much. Do you have time for questions, or does anyone have any question or anything else to say? The few people who are here, you have time for questions?
Yeah, of course we have time for questions, and
Yes.
we'd also like to know, because well, we have to be honest. We look at this from a more. We know this is a global problem, sickle cell disease and beta thalassemia, but we're looking at it as a locally, like we have 600 patients in the Netherlands, or 600 in Rotterdam, 1,500 in the Netherlands. But for us, it would be very useful to hear from the perspective in countries where you really have large numbers of patients.
Yes.
Because here it's called a rare disease here, and it is. It fulfills the criteria for a rare disease. But of course, I know in some countries in Africa, you get tens of thousands newborns every year. And that is numbers that we cannot even begin to understand. So we'd like to hear that side of the story as well.
Okay. I'm a biochemist by training, and incidentally, I did a literature review a couple of decades ago on abnormal hemoglobins. And I am, my genotype is AA, just to let you know how common and frequent this is in Africa. And that your data that says about 5 million, I think it's conservative. We'll probably have more than 5 million in Africa that have sickle cell. And also carriers will be more than even the, is it 80 million or 50 million or so that you mentioned, Professor Borg. I have 3 in my family, and 2 of my children, as well as my wife, are carriers, okay? And it's a serious thing here in Africa. Before you get married, one of the things they do is you must be tested for the sickling gene. It's a must before you get married. I have seen people who have ignored that advice, and out of 4 kids, unfortunately, all 4 kids were, sickle cell kids. And over a space of 8 years, all the kids died. The stress, the trauma is really bad. So it's a big issue here. We have a sickle cell research center in one of our teaching hospitals here in Lagos, and I know they built one somewhere in Ogun State, and I'm sure there are quite a lot around. There's even an association of those who are doing research on sickle cell. A couple of them are supposed to even be on this meeting. This is why I'm thinking they might want to see the presentation, and I'm sure they will be excited. So the question I want to ask is a question I was hoping some of these guys may ask. To date, I'm not sure how, since I'm not in clinical research or in drug dispensing, I wouldn't really know, but it'd be good to know that what, percentage of the physicians are aware of the current research that is going on in sickle cell, as well as what is being used. So to date, like Professor Borg mentioned, hydroxyurea is being used. It's been approved since 1998 from what I heard, and it's the one that is supposed to be used. But even then, for some reason, it is not being used as much as it should be, perhaps because of some of the side effects. So what would you say is the novelty in those molecules or the chemotypes you're talking about? What makes them different? What advantage do they have over hydroxyurea?
Exactly.
Yes, so hydroxyurea is, well, you're a biochemist, so it was initially developed as a chemotherapeutic drug.
Yes.
And it's a cell cycle inhibitor, and that is why people are always concerned that it might also have genotoxic effects, and that doesn't appear to be the case. On the other hand, there's a large number of patients with sickle cell disease that do not show a very good response, and we have actually performed a study where we looked into this by taking the extremes of the good and the poor responders. And we found in that study, it was relatively small, that really depended on the baseline level of fetal hemoglobin in the patient. So if that was already fairly high, then you could double it, and then you go above a critical threshold. So then it is good. But if you start out low, well, you would still double or even treble it, but then it would not be high enough. So what would we be hoping to achieve? Of course, we do not know what the compound is at the moment, but that it is a compound that is much more specific than hydroxyurea, which is very generic, of course. Yeah. It will have an effect on all the cells, and also is much more specific for the induction of the fetal hemoglobin. And that's why it's so important that we know all the molecular details of the repression mechanism. And we know, for instance, this BCL11A repressor protein in a red cell. It has this particular function, so repress the fetal hemoglobin genes, but it does very little else. So that is a very good target, and we understand that. So this protein doesn't work by itself, and we know about the network of proteins that it works together with, and this is what we are focusing on. So in that way, that we will be much more specific than is possible with current treatments, and more effective.
So if you like, hydroxyurea will act as a form of a benchmark that anything that we identify needs to be as good or better.
Better than it.
It needs to be better.
That's how it is.
Yeah,
exactly. That's what I was hoping to hear. And I'm hoping people will hear, the professionals will hear, and they will be interested in providing as much collaboration as they can in this area where this disease is endemic, to help people in this research. It's being a target molecule. I mean, it's to target a specific gene, unlike the wide application or activity of the hydroxyurea. So that is really a key factor in this research. And from my point of view, and I'm sure all people who are in that area, it's truly a novel approach. And I'm hoping that your research progresses very, very well.
Okay.
So if I may ask a question, because you mentioned genetic testing, and I know in some countries, for instance, I was in Iran, and I was surprised to learn that it is compulsory, the testing, and there they have beta thalassemia. But every couple that wants to get married, they have to be tested. And we know from Europe, from Malta, Cyprus, Italy, and Spain also. I think that the testing programs, the genetic testing programs, were very, very successful. And like in Cyprus, they didn't have a single new case of thalassemia for 20, 30 years. And
Wow.
So yeah, but now the young people, they forget it. They don't know the disease. So they think, oh well, I'll just take. So now they get in case again. But so how common is genetic testing in Africa? Because that could do a lot of good as well.
Okay, when you say genetic testing, you're referring to simply testing using the blood, right?
Yeah, you could do that. You could do that.
Okay, that's actually what is very common here, and is very common, easy to do. That labs everywhere that will do your genotype tests. What they are beginning to advocate now is to begin to do it from for infants, for neonates.
Yeah.
In fact, lady currently on this Zoom meeting with us, Mrs. Akimbo, the Nurses Association in Nigeria had a Zoom presentation 2 days ago on the concept of promoting and pushing for testing of neonates for sickle cell. I almost sent that invitation to Professor Borg, but it was just the Nurses Association. And it's because I'm married to one that I became, to that. But it's something that has just been done. But as per the genotype, it's already decades ago now. It's already something that is well known. But like you said, many people, especially in the middle class or literate, semi-literate to illiterate people, and level of literacy in Africa is still quite high, still are reluctant to do so. Many people just don't want to do it. The reason is that they've not been doing it from when someone is a child or from newborn, which is important. Data, keeping of data and databases are things that are just coming up in Africa. So to have a database of people so that you have your blood record or your genotype and stuff, people often don't know until they are in their college days, or they are planning to get married, or they need to get some blood transfusion or do some blood work. Even in the routine medical tests that they do in the hospitals, they don't often include genotype. But now, when you want to enter universities, they will require that you do your genotype. So most people will know their blood group, their genotype, and co. So when they want to get married, most people are now trying to find out. I personally know so many people who were planning to get married, and a year to the time they went to do their genotype, and they found both of them were carriers. And it was a difficult decision for them to break up. And I've known cases of young men and women who decided to go ahead and get married, believing that, there's a 50% chance we will have a normal child and a 25% chance, maybe we will risk it. And unfortunately, some of them didn't get good results. So we need more awareness. People need to get more information. But there is enough testing already available, and they're now pushing for neonatal testing, newborns, testing for that.
So here it is part of, you might be surprised to learn, but here it is part of neonatal testing since about 10 years. So they also test for cystic fibrosis, phenylketonuria, and so they have, I don't know, maybe about 20 diseases, but the sickle cell disease is part of the test, and the most important factor for that is that they now know the carriers from a very young age.
And that's important.
The patients, you find the patients within the course of the first year, but now they know the carriers, and so I do have good hope that we will at least here be able to control this, at least in the future. I should also say that even not even here, but with the best clinical care that we can provide, we still lose babies, teenagers with sickle cell disease. It's just unbelievable. Like I have some pictures like in our pediatric hospital, and there was the director of the Feyenoord football club. He was presenting a check with money. and there was once little baby there, and so I sometimes use it to show that, you know, we're fundraising and we really investigating this disease. And it turns out that the baby had just died like 3 months later, and this, so it's devastating. But I am asking about the genetic testing, because that of course is we should not ignore that, because if you really want to do something in the short term, then that is where the road, that is the quickest way. It's preventing, you get more testing. So I'm very glad to hear that this is at least in place, and then of course it could be better, that is obvious. But that is very good to hear.
I hope that maybe through this medium, we can be useful. AICTF can help connect you with people who are researching along those lines, or who are working with patients, to provide whatever information or, you know, whatever it is you might be needed for you to understand the peculiarities. You said it's pleomorphic, frankly, and the way it presents here, even though the mechanism of action, the expression is the same, but the way it presents and maybe regional or genetic differences, if you can also understand that, it can help with what you're doing to come out with something that is more global in approach.
Yes. Okay.
Thank you so much.
Yes, absolutely.
Okay. So I think this brings the meeting to an end.
Yes.
We stop here. Okay. Thank you. Thank you for hosting us.
Thank you very much.
Thank you.
Laura for
Thank
joining, and Jacques for making his presence as well.
I enjoyed it very much. So let's let's say, well, let's keep in touch.
Yes, sir. We would like to do that. Thank you so much. There's someone.
Okay.
All right. Thank you.
Bye-bye.
Thank you, Prof.
Okay,
Thank you,
bye-bye.
Jack.
Bye-bye.
Yeah. Thank you, sir.
Okay.
Say again?
Thank you very much.
Okay.